Okay. Hello, my name is Carly's Theron. I'm from Group 2, and the pathogen we're presenting on is HIV. So let's get started with the origins and impact. Approximately 1 century ago in southeastern Cameroon, cross-species transmission of the simian immunodeficiency virus from chimpanzees gave rise to HIV-1 group M, the principal cause of the AIDS pandemic in humans. In 1968, Robert Rayford was the first AIDS patient recorded in the United States, whom admitted himself to a hospital in St. Louis, Missouri, as he had been coping with a dangerously low immune system, inflammation, plus other life-threatening complications. Now, in 1981, the CDC releases a report documenting instances of uncommon lung infections. It was identified when a rising number of young homosexual men began experiencing uncommon opportunistic infections and rare cancers. Next slide. The virus was identified in the early 80s by three doctors, two from France and one from the United States. Initially, the virus was referred to as lymphadenopathy-associated virus before being officially named HIV. Next slide, please. So what is HIV? HIV stands for Human Immunodeficiency Virus. It specifically targets the CD4 cells and affects the body's immune system. If untreated, HIV can progress to AIDS, which is acquired immunodeficiency syndrome, where the immune system is severely compromised. Next slide, please. So let's talk about the epidemic. HIV/AIDS quickly expanded beyond initial groups and regions due to factors like travel, migration, and sexual transmission. Next slide, please. So, understanding the epidemiology of HIV is crucial for developing targeted interventions and policies to mitigate the spread of the virus. According to the World Health Organization, we can see that in 2022, more than 39 million people were living with HIV globally, with over 50% being in Africa. And according to this data, we can assume that probably 2 in 6 people may acquire HIV. And according to the data, 630,000 people had died from HIV. Taryn, it's on you.
Yeah, hi, I'm Taryn Collins, part of Group 2. I'm going to be talking about the transmission of HIV. It's primarily transmitted through unprotected vaginal and anal sex with an infected partner. These activities can lead to the exchange of bodily fluids containing the virus. Another way of transmitting the disease is by sharing needles and drug paraphernalia, such as syringes and cookers. Among these, intravenous drug users are um, transmitting it through contaminated equipment, and this can introduce the virus directly into the bloodstream. Additionally, perinatal transmission or mother to child transmission can occur during pregnancy, childbirth, or even during breastfeeding. The virus is passed from an infected mother to her baby. It's crucial to also note that HIV does not survive long outside of the human body and cannot reproduce outside a human host, meaning transmission through things like insect bites, saliva, tears, even the air or our sweat is not possible. Uh, Next, we're going to talk about the symptoms of HIV. There are 3 stages. In the early stages of HIV infection, individuals may experience flu-like symptoms such as fever, fatigue, sore throat, lymph nodes are swollen at this point too. As the infection progresses to the chronic stages, symptoms might subside and the virus does become latent. This is when the virus does continue to replicate, gradually weakening the immune system. Without treatment, HIV can progress into what we call AIDS, characterized by severe symptoms including rapid weight loss, recurring fever, chronic diarrhea, and at this time can turn into susceptible, life-threatening illnesses. Lastly, we're going to talk about the diagnostics of HIV. So the CDC does recommend that everyone between the ages of 13 and 64 get tested for HIV at least once as a part of routine healthcare. For people with certain risk factors, the CDC does recommend getting tested at least once a year. Um, Diagnostic tools included for testing for HIV are antibody tests, antigen antibody tests, and then also NATs. NAT stands for nucleic acid tests, which directly detect genetic material of the HIV virus, providing the most sensitive and accurate diagnosis. However, things like antibody tests, which is where they detect antibodies produced by the immune system in response to the virus, that's more of the initial screening. There's also antibody antigen tests that detect both HIV antibodies and antigens and proteins produced by the virus itself. So, yeah.
Okay, moving on to the molecular biology of HIV, the viral envelope surrounds the nucleus, protecting the RNA and proteins, and consists of glycoproteins GP120 and GP41. The HIV envelope glycoproteins play an essential role in the virus replication cycle by mediating the fusion between viral and cellular membranes during the entry process. The capsid acts as a safeguard to the viral RNA and protects it from host cell enzymes as well as immune defenses. The capsid also plays an important role in delivering the viral RNA into the host cell cytoplasm and helps in the replication of HIV virions. HIV enzymes are integral to HIV's life cycle and play important roles in the different stages of infection. The first stage of the HIV life cycle is the binding stage, and this is when the GP120 that is found on the viral envelope identifies and interacts with CD4 receptors found on the surface of the host cell. CD4 receptors are found on macrophages and T cells, and this is why these are the cells that HIV targets. The binding of GP120 to the CD4 receptor then exposes the co-receptors CCR5 in macrophages and CXCR4 in T cells. GP41 then allows for the binding of GP120 with the co-receptor of the host cell, which then leads to the fusion of the viral envelope with the host cell's membrane and allows for entry of the viral core into the host cell cytoplasm. Once the viral core is inside the CD4 cell, the viral protein reverse transcriptase converts the viral RNA into DNA and enters the host cell's nucleus. The new strand of viral DNA is then integrated into the existing DNA of the host cell's genome with the help of the HIV protein integrase. The completion of this stage is what marks lifelong infection of the host. Once HIV is integrated into the host cell DNA, the virus begins to use the machinery of the CD4 cell to create long chains of HIV proteins. The protein chains are the building blocks for more HIV. Along with the protein chains, envelope proteins are also created by the host cell. So lastly, the proteins that were just created by the host cell are then transported to the host cell membrane, and they form a non-infectious HIV virus. Once assembled, the immature HIV buds from the surface of the infected host cell membrane, and the HIV protein protease then begins breaking down the protein chains inside the immature HIV, which results in structural rearrangements within the virus and results in mature HIV that is capable of infecting other cells. Moving on to Erica.
All right, on to current treatment. As of today, there is no cure for HIV. However, the standard of treatment when diagnosed with HIV is called the highly active antiretroviral therapy, also known as HAART. This uses a combination of antiretroviral drugs, and once a patient is on it, they cannot stop, so it is a lifetime treatment. They all inhibit HIV replication at different stages through different mechanisms, and they are co-administered to prevent drug resistance from occurring. There are 5 types of antiretroviral drugs, which are the entry inhibitors, nucleoside reverse transcriptase, non-nucleoside reverse transcriptase, integrase strand transfer inhibitors, and pharmacologically boosted protease inhibitors. Drug resistance can occur, which requires changes in active drugs. However, virological failure due to drug resistance is very rare. On to prognosis. All right, so the prognosis for HIV can be very positive or negative. If left untreated, HIV may develop into stage 3, which is also known as AIDS. It can take about 2 to 15 years to get to stage 3. However, once it gets to stage 3, though, the life expectancy is only about 3 years. However, if detected and treated at stage 1 or 2, patients can live a very normal life by being consistent on HAART. Through this treatment, the infection stops progressing to the point that it no longer damages the immune system and it cannot be transmittable. All right, on to the challenges. So there are quite a few challenges people face with HIV. One is the stigma. Social stigma such as negative attitudes and prejudice with people with HIV occur because there's a lot of misconceptions and misinformation around. This can cause a reluctance to seek medical help. There is also a limited access to healthcare around the world. This is caused by many different reasons, such as lack of transportation, lack of healthcare, limited availability of specialized HIV care, and etc. All right, there's also a few challenges in developing a vaccine. High rates of mutation and recombination during viral replication makes it very difficult to make a one-fits-all vaccine. There is also a lack of investments from pharmaceutical companies, which prevents researchers getting enough money to continue on developing a vaccine. And lastly, the lack of animal model makes it very difficult to test the developing vaccine as well. And so this was Group 2's presentation on HIV, and I hope you learned something new or valuable today. Thank you.
Thank you.